In this occasion, I'm going to talk with you about the inflammatory response and obesity, a pretext to talk about the risk factor for the development of endothelial disease and its consequences. The objective is to involve you in the integrative thinking of a sequence of events that, as a chain reaction, are triggered one after the other, conducting from the oxidative stress to alter metabolism, followed by the presence of atherosclerosis and other pathologies related to the chronic inflammatory process. Inflammatory response is always linked to many elements involved, as slavons beginning for oxidative stress, methylation of genes, metabolic modification, and continuing with endothelial disturbances related to the most important causes of death, cardiovascular diseases. This is the risk factor for the oxidative stress and inflammatory response. And I put an additional with tobacco and sedentary attitude in the life. Obesity in the past was equal to long-term positive balance, a matter of energy consumption, caloric expenditure, fat storage, but the answer is not as simple as this. And we can see the elements of the inflammatory process in obesity in this slide, beginning with oxidative stress, inflammatory response, immunologic activity, innate and adaptive, with epigenetic modifications that alter the genes and RNA transcription, disrupting proteins production and changing the metabolism, with complications in the phenotypic and physiologic disturbances, with the structural modification in the endothelium, with the production of atheromas. The brain is the headquarters for the control of hunger and satiety, energy expenditure, and the regulation of growing and reproduction, and has a tightly relation with adipose tissue and the production of leptin, with the pancreas and insulin, peptide Y and CCK in the bowel, and hormone ghrelin in the stomach. Talking about factors that precipitate in obesity, modernization promotes increased calories intake and decreased physical activity. Genetics contribute to 40 to 70% of obesity, with more than 50 genes that are strongly associated with obesity. Presence or absence of genetic factors protect us or predispose us to obesity. 19 years of research in obesity have discovered the existence of genetic continuum between monogenic and polygenic, which points to the role of genes involved in central regulation of food intake and the genetic predisposition to obesity. 9 loci are recognized to be involved in Mendelian forms of obesity, along with 58 loci that contribute to polygenic obesity. Genome-wide associated studies identified 36 polymorphisms associated with abnormal body mass index and identified variants explaining 1.45% of the variance in body mass index. Prevalence of monogenic obesity can be estimated between 5 and 10%. In genetic terms, predisposed subgroups are particularly vulnerable to obesity in obesogenic societies with unlimited access to food. Genome and epigenetics are very important. Express alteration within the DNA sequence, but we suppose it is going to persist in passing the time. It includes DNA methylation, non-coded RNA, histone modification, chromatin remodeling, and has an important role in the regulation of tissue-specific gene expression. It can induce long-term change in genes function and metabolism persisting through an individual lifetime and can be inherited from generation to generation, and we suppose it could be in 4 generations. DNA methylation generates decreased activity in mitogenic protein kinase, changing the expenditure of energy. Insulin signaling blockage generating diabetes, inhibition of signaling of calcium channels and producing hypertension, change some genes in skeletal muscles and modifying the production of energy, obtaining as a final result change in metabolic process. Epigenetic patterns are established in any moment of life and under the influence of risk factors, especially in the period of extreme plasticity, as is the perinatal. Genes methylation can persist for all life long, preparing the human body for the risk of food deprivation, climatic change, or a toxic environment. The methylation change can predispose in the future to the fetus suffer from obesity and diabetes type 2, among other disorders. Analyzing DNA in skeletal muscle in 2 million samples, researchers found 603 methylate genes or switch-off system in diabetes type 2 and metabolic syndrome unable to signal. Oxidative stress is the beginning of a chain reaction, refers to elevate intracellular levels of reactive oxygen species that causes damage to lipoproteins and methylates DNA. It is associated with a large number of pathologies. In this figure, we can see the increase of reactive oxygen species in the environment of obesity and the presence of white adipose tissue, increasing oxidative stress to remote tissues, and dysregulating the production of adipose cytokines, increasing insulin resistance, diabetes, atherosclerosis, and metabolic syndrome. Inflammatory process in obesity has 5 participants or protagonists: adipocytes, preadipocytes, dendritic cells, T cells, and macrophages. Participating with the production of interleukin, leptin, and adiponectin, monocyte chemoattractive factor, spillage of free fatty acids, increasing of the cytotoxic T lymphocytes, and augmenting the pro-inflammatory macrophages type 1. Inflammasome, a multiprotein oligomer of the innate immune system in cytosol, is responsible for the activation of inflammatory activity that promotes proteolysis, maturation, and secretion of pro-inflammatory cytokines as interleukin-1 beta and interleukin-18, as well as cleavage of gasdermin D, whose induced pro-inflammatory program cell death termed pyroptosis. The dysregulation of activated inflammasome can trigger diseases such as cancer, autoimmune, metabolic, and neurodegenerative. Inflammasome has 2 phases. In the first hit, increase the production of interleukins, inhibitory monocytes factor, tumoral necrosis factor, and pro-interleukin-1 beta. And in the second hit, or second phase, with increase of caspase-1, the increase of interleukin-1 beta. Remember that caspase is involved too in the production of inflammatory storm and multi-organ failure. Adipogenesis and pro-inflammatory environment increase pre-adipocytes and increase the production of interleukins and monocytes inhibiting factor, macrophages and T cells polarization, and pro-inflammatory environment decreasing phosphorylation and decreasing translocation of GLUT4, the transporter of glucose, increasing insulin resistance, and the presence of diabetes. Neuroendocrine system has a big door open in the blood-brain barrier, to establish an interrelation between hormones and peptides with the nucleus arcuatus neurons in the hypothalamus. Ghrelin, peptide Y, leptin, insulin, peptide OXM, GLP-1, and CCK are interrelated in a feedback system with the nucleus arcuatus. Dysregulated activity of arcuatus neurons due to inflammation is responsible for excessive consumption of calories and the onset of obesity. co-express diorexigenic neuropeptides and co-express anorexigenic peptides. Two distinct types of adipose tissue, brown and white, in the biochemistry of obesity. Brown is associated with thermogenesis, control of adiposity, and inversely correlates with body mass index. The most important reservoir for obesity and insulin resistance is white adipose tissue that also stores triglycerides from an excess of energy, mobilized reserves during nutrient deprivation, and acts as an endocrine organ releasing adipokines, including interleukin 6, beta, tumoral necrosis factor, leptin, and adiponectin. Adipocytes induce molecules, adhesion molecules in the denudate endothelium and attract monocytes to the endothelial region and transform them into macrophages that swallow oxidative LDLs, forming foam cells and the atheroma plaque. These are participants of the inflammatory process in obesity: pre-adipocytes, macrophages, T cells, dendritic cells, natural killer cells, eosinophils, bone Kupffer cells, and skeletal muscles. Some participating with the presentation of antigens, others producing inhibiting factors and chemotactic factors, interleukins, others activating the immune response, adaptive and innate, and the others prolonging the inflammatory process. This diagram tries to express the complexity of the inflammatory process in the presence of obesity, with increase of oxidative stress, the production of reactive species of oxygen, and the increase of caspase-1, the presenting of molecules associated to damage pathogens, and pathogenic associated molecules of pathogens, decreasing the receptivity of the insulin receptors and decreasing translocation of glucose-4, modifying the intermediate metabolism of lipids, proteins, and glucose, prolonging the inflammatory process and creating a chronic inflammatory response, promoting Alzheimer's disease, atherosclerosis, autoimmune disease, cryopyrin-associated process, changes in the tissue repair, and in the production of tumors, and type 2 diabetes. Obesity is associated with reduced physiologic response to leptin and insulin, leading to the concept of obesity-associated hormonal resistance. Activate by saturated fatty acids and pro-inflammatory cytokines that promotes the release of inflammatory mediators. Constant activation of leptin receptor neurons in aqueous nucleus leads to the glucose intolerance. Caspase-1, which alters phosphorylation, triggers insulin resistance and reduces glucose metabolism. Inflammation is a critical mediator in obesity. Initiates chain reaction, accompanies endothelial disease, which is the protagonist of the #1 cause of global death by cardiovascular complications. Como ustedes podrán ver, el proceso inflamatorio en la obesidad es muy importante. Hay una secuencia de eventos que se repite cada que tenemos factores de riesgo asociados. Estos factores de riesgo definitivamente modifican el comportamiento del endotelio y es lo que al final va a generar la causa de muerte más importante en el 85% de la población occidental, que son las enfermedades cardiovasculares. Aquí trato de explicar de una manera sencilla y simple cómo se van modificando los comportamientos bioquímicos con cada uno de los factores de riesgo. Sin embargo, cualquiera de ellos o todos ellos en conjunto pueden tener el mismo comportamiento y llevar al paciente a la debacle con una enfermedad cardiovascular, un embolismo, un infarto al miocardio o un infarto cerebral. Agregaría yo otro factor de riesgo después de escuchar la plática del doctor Tarikul, que es precisamente el estrés, que el estrés puede simular prácticamente cualquiera de los factores de riesgo y sus comportamientos. Y bueno, pues este es el mensaje que yo quería transmitirles. En segundo lugar, quiero dar las gracias a la doctora Claudia Arias, presidenta del 36º World Congress of Internal Medicine, to the Dominican Republic Society of Internal Medicine, the Dominican Association of Internal Medicine, and the local medical associations, societies, and colleges from the Dominican Republic for such a spectacular academic event. Thank you so much. And International Society of Internal Medicine invites you to the 37th World Congress of Internal Medicine, hosted in Prague in 2024. Thank you very much.